Hello friends, good morning. So, uh, in this lecture, we are going to learn about the Mendel's work. So, Mendel set the plant hybridization experiment. So, for our convenience, I have divided the hybridization experiment in two parts. So today I will be discussing about the monohybrid cross. In the next lecture I will be discussing about the dihybrid cross. So let us start with the Mendel's work. So before starting Mendel's work, first of all I want to tell you that Mendel derived three ideas from his work, and that three ideas become the backbone of principle of inheritance. So what were those three ideas? The first one was dominance, then segregation, and lastly the independent assortment. So in later slides we'll know that first two idea was derived from the monohybrid cross, and for getting the idea of third, that is independent assortment, Mendel conducted the dihybrid experiment. So let us start with the monohybrid cross. So in our modern understanding of how trait may be inherited through generations come from the principles proposed by Gregor Mendel in 1865. We have already seen in the last class that Mendel started his work or completed his work in 1865, but he published his work in 1866. After 8 years of tedious experiment with these plants, and Mendel admitted by his own that some courage was needed to persist with them, Mendel proposed the three foundational principles of inheritance. So these three principles in the coming lecture on this in this lecture, also in coming slides we will see what were those three foundation principles of inheritance. So regarding the principles of inheritance, we already know about the principle of inheritance proposed by Mendel. You might have studied it in your +2 classes, but here in this lecture I will be discussing it into slightly different way, but the thing will be same what you most of you or all of you is knowing, but today I will approach it in slightly different way. So Mendel was curious about how trait transferred from one generation to next. So for this purpose he set the experiment, and after setting out the experiment he understood the principle of heredity in mid 1860s. So he chose, I have already discussed in the last class that he was working initially with two plants, but finally he concentrated on the pea. So why pea plant acted as good model system? Because in pea we can control the fertilization by transferring pollen with a small paintbrush. So here you can, in this picture you can see that with a small paintbrush you can transfer pollen from male to female part of the same flower or of the other flower with the help of paintbrush. So the pollen come from the same flower, it is called as self fertilization, and if it come from another plant's flower, then it is called as cross fertilization. So this was possible in case of pea plant with the help of a small paintbrush. Now the first Mendel observed plant forms and their offspring for 2 years. So what he did, he carefully observed the pea plants for 2 years as they self-fertilize. So he started plant self-fertilization and observed that plant for 2 years, and he ensured that their outward measurable characteristics, that is phenotype of particular trait, remained constant in each generation. So he self-fertilized the flowers and he observed the traits, outward outwardly expressed trait, that is phenotype, in each generation. During this time, Mendel observed 7 different characteristics. So what were those 7 characteristics? Here you can see it was plant height, flower color, flower position, pod color, pod shape, seed color and seed shape. So in plant height, tall on dwarf, flower color purple white, flower position axial and terminal, pod color it was green and yellow, pod shape inflated and constricted or smooth and constricted, seed color yellow and green, and seed shape round and wrinkled. So these 7 traits he studied, and the characteristics include the height, we have already seen. Now Mendel spent years letting the plant self-fertilize. We already seen in the last slide. He verified the purity of his plants by confirming that tall plant had only tall children and grandchildren and so forth. So he self-fertilized the plants, studied it for generation, and he ensured that the tall plant is having tall progeny and it is having tall grand progeny and so forth. Because the 7 pea plant characteristics tracked by Mendel were consistent in generation after generation of self-fertilization, these parental lines of pea could be considered as pure breed. So those plants who show consistent character, consistent for a particular trait over the generation, Mendel considered it as a pure breeder, or in modern terminology, homozygous. Now, by this way, Mendel developed 22 varieties of pea plants. 22 varieties of pea plant, that is tall, dwarf, this color, that color, variation of seed shape. So like that he developed 22 varieties of pea plants. Mendel do not crossed pure breeding parent, but he also crossed hybrid generation. Mendel not only crossed the pure breeding parents. So what he did, first he did self-fertilization and ensured that they are true breeders or homozygous. And after that he not only crossed the pure breeding plants, but he self-hybridized the hybrid progeny. That means he developed the F1 by crossing 2 parents of 2 different 2 different form of a trait, and he self-fertilized the F1 plant. So these crosses, which in modern terminology are referred as F1 reciprocal. So you might be knowing F1 means parent 1 crossed with parent 2, parent 1 crossed with parent 2 and produces F1. Then what is F1 reciprocal? So initially suppose parent 1 was male and parent 2 was female. In the latter experiment, if you want to prepare the F1 reciprocal, you have to take parent 1 as female and crossed with parent 2 male, and the progeny produced F1 will be called as F1 reciprocal of what? The earlier cross, reciprocal of earlier cross. That means the sex of parents will be interchanged if you want to prepare the reciprocal. Similarly, he crossed B1, that is back cross with first parent, then back cross with second parent, that is B2, are the classic crosses to generate genetically hybrid generation. So he developed the hybrid generation by developing such type of crosses or performing such crosses. Now you see, breeding white flower and 2 breeding purple flowers, the result was a hybrid offspring. So when he crossed white flower with purple flower, the result was the hybrid offspring. So rather than being mix of 2 colors, so what we can expect, if we are crossing the white flower with purple flower, we can expect that the F1 progeny or the hybrid progeny will have intermediate phenotype or intermediate color of the flower. But instead of that, rather than being mix of two colors, the offspring was purple flower. So instead of that, he observed that hybrid plant was having purple flower. It was neither white nor the mix of two, white or purple. So from this, Mendel conceived the idea of heredity units, which he at that time he called it as factor, and now we know that those factors are called as genes. So he conceived the idea of hereditary unit, which he called factors, and he further went up to saying that one of which is recessive characteristic and other is dominant. So based on that, based on observation of F1, he conceived the idea of hereditary units, and he told that some of the traits are dominant and another is recessive. Mendel said them factors, later called genes, normally occurs in pairs in ordinary body cell, yet segregate during the formation of sex cell. So what are those genes? Those genes are normally occurring in body in pairs. So I will tell you in the later slides how they are arranged, where they are present in the cells or body. So and after that they will segregate during the formation of sex cell. So before proceeding further, if you see the previous slide, here in this photograph you can see that he removed stamen from the purple flower, then transferred pollen from stamen of white flower to carpel of purple flower, then pollinated carpel matured into pods, planted seed from pod, examined offspring, and he found that they all were having purple flower. So he crossed white flower with purple flower. In F1 generation he got all the purple flowers. So whatever we have studied, just this is in the summary form in this picture. Now each member of the pair become part of separate sex cells. So what we earlier seen that the factors or genes, they are present in pair in the body or in the cell, and each member of the pair become part of the separate sex cell at the time of formation of sex cell. Each member of the pair will come in a sex cell. That means in sex cell that pair will break, and in sex cell there will be only one copy of that. So the dominant gene, such as the purple flower in Mendel's plant, will hide the recessive gene, the white flower. So what Mendel observed, that dominant gene was purple because it was hiding the expression of white flower. Then after that what Mendel did, he self-fertilized the F1. So here you can see that he self-fertilized the F1, that is hybrid purple flower, F1 generation, and obtained the F2 generation in 3:1 ratio. So here he got the F2 generation, and here you can see the number, the actual experiment performed by Mendel. He got 705 violet flower or purple flower and 224 white flowers. So that means these flowers are in the ratio of 3:1. So in the F2 generation he got flowers in the ratio of phenotypic expression, phenotypically in the ratio of 3:1. He correctly theorized that genes can be paired in three different ways for each trait, that is capital A, small a, and capital A, small a. So whatever experiment he did, he noted, actually in the last class also I have discussed that he conduct the experiment, he made a meticulous note of that experiment. That means his record keeping was very perfect. So what he theorized, that each of the character is controlled by gene, and gene can be paired in three different ways. that is capital A, capital small a, and capital A, small a. The capital A represents the dominant factor, that is dominant gene, while the lowercase a, that is small a, represents the recessive one. So from here, after getting F2 result in the ratio of 3:1, he proposed that the character can be, or the genes can be paired in 3 different ways if he is considering only single trait. That is capital A, if capital A is represented for dominant gene and small a for recessive gene, then it can be paired in 3 different ways: capital A combined with capital A combined with small a, and small a combined with small a. Now this you can understand it by Punnett Square. So this Punnett Square was given by Punnett, that is Reginald Punnett was a scientist who proposed, or he gave a square which can be used for different combination of alleles or genes. So this is used to predict the possible offspring genotype, how these alleles or genes are combining, and that will form the genotype. The place alleles for each parent of each side. So either side you write male gametes and female gametes. So suppose the mother's genotype was capital W, small w, and father's genotype again capital W, small w. So on mother side what will be the genes or alleles? It will be capital W, small w. Similarly on father side it will be capital W and it will be small w. Now the combination you can see here, capital W combined with capital W, that is capital W, it will be white flower, white. Then purple flower. Then capital W combined with a small w, it will again be purple flower. Again here capital W combined with a small w, capital W, small w, this small w, this capital W. So this will again be purple flower. Then this small w, small w. So this will be the small w, that will be the white flower. And here you can see the ratio is 3:1. So by this way, with the help of Punnett Square, we can see the combination of male and female gametes, and we can easily keep the record of how the progenies are produced and in what ratio these progenies are produced. So if you see the phenotypically, the 3 purple flower for 1 white flower, and if you see genotypically, the 1 purple flower is dominant homozygous, another 2 purple flower they are heterozygous, and the 1 homozygous for recessive. So genotypic ratio will be 1:2:1. Then next, based on this, Mendel proposed the law of segregation. So what is law of segregation? He observed that each individual has 2 alleles. So we have already seen that each individual is composed of 2 alleles. In later slides we will see how these alleles are present, where these alleles are present. So each individual has 2 alleles for each trait, 1 from each parent. So each allele is coming from each of the parents. Suppose I am having 2 alleles, so my 1 allele may be coming from my mother, another allele may be coming from my father. Thus he formed the first rule, that is law of segregation. So based on this observation, he proposed the law of segregation. What is law of segregation? He told that individual possesses 2 alleles and a parent passes only 1 allele to his or her offspring. So each individual is having 2 allele. I am having 2 allele for a particular trait. That 1 allele has come from my mother, another allele has come from my father. When my progeny will come, I will be providing my progeny only one allele, not two allele. One allele I will be providing to my progeny, other allele will be coming to my progeny from my wife. So, in this way, the Mendel proposed the law of segregation, that is, individual possess two alleles. I am possessing two alleles, but I am passing only one allele to my progeny. That means these two alleles will be segregated. So, allele pairs separate or segregate during gamete formation and randomly unite at fertilization. So, what it states that I am having two allele, but I will be passing to my progeny only one allele. That means at the time of gamete formation, allele will be segregated and only one allele will be going into the sex cell or gametes. And another allele will be uniting randomly at the time of fertilization, whatever allele is coming from other parent. One allele is given by female parent and the other is given by male parent. The two factors may or may not contain the same information. So it is quite obvious that whatever allele I am having, it may contain information that may be similar to the information that is coming from allele of my wife, or it may be dissimilar. That is totally independent thing. If the two alleles are identical, these are called as homozygous. Okay, if same allele is coming, same type of allele with same information, if it is coming from my wife to my progeny, then these two together called as homozygous. And if two alleles are different, then it is called as heterozygous. So this is the concept of homozygous and heterozygous. Now, you see the arrangement of alleles at homologous chromosome. Suppose this is my cell. This is the cells of my means chromosomes from my cell. So these two are the homologous chromosomes, homologous pair of chromosomes. That means one chromosome has come from my mother, another from my father. So this is the allele for purple flower. Suppose this is allele for purple flower. Suppose this is the chromosome for pea plant. So this has come from the mother of pea plant and this has come from father of pea plant. So this is the allele, one allele from mother and another allele from father. Here you can see that based on color, this is pink color, just for depiction. Deep pink is light pink. That means these two are different. So these are the heterozygous. They are having different information. So locus for flower color gene, allele for purple flower, allele for white flower. So these can be considered as heterozygous. So these alternative form of a gene that code for a trait are called alleles. There are two alleles for each trait, one allele of a trait from mom and another one allele from dad. So now you can understand how alleles are arranged on chromosomes. So in the where alleles are present in the cell, in nucleus, on chromosomes. Now, so first law, we see that Mendel has proposed first law of segregation, which is considered to be the universal law. That law is having no exception. Another phenomena was proposed by Mendel, that is known as phenomena of dominance. Many people, or in many books, it has been written that it is law of dominance. But as per my view and many other books also, it has been mentioned that it cannot be the law because this law is having many exceptions. That is why I prefer to call it as a phenomena of dominance. Phenomena of dominance, not law of dominance. So, what this phenomena of dominance states, the presence of an allele does not promise that the trait will be expressed in the individual that possesses it. So, suppose I am having a particular allele, it is not guaranteed that that allele will be expressed in me. It only depends that what other allele I am having. Because you know that trait or a gene is having two alleles. So even if one particular allele I am having, it is not guaranteed that allele will be expressed or not. So in heterozygous individual, the only allele that is expressed is called as dominant. The allele which is dominant, that allele only will be expressed. The recessive allele is present, but its expression is hidden. So suppose I am having one copy of allele that is recessive. So that allele cannot be expressed because it will be hidden by the expression of dominant allele. So the genotype of an individual is made up of many alleles it possesses. Here you can see that the capital A, here the fruit color you can say it is red. If it is small a that is recessive, then the fruit color is yellow. Now in case of heterozygous capital A small a, here you can see the fruit color is red. That means the capital A red color is dominant over the yellow color small a. So even if small a is present, it is not guaranteed that it will be expressed. For expression of small a or yellow color, you need to have both the copies of allele as small a, that is allele for yellow color. So an individual physical appearance or phenotype is determined by its allele as well as by its environment. So this is a different thing, but because if it wanted to tell that what is phenotype. Phenotype is the combination of genotype and environment. In the latter part of this unit, we'll be seeing how this genotype is affected by environment. So as of now, you just remember that how dominant and recessives are being expressed. Now, most of the animals have paired chromosome and are diploid. They have two versions of each chromosome by female parent in ovum. Two versions of each chromosome, that is homologous chromosome. One is by female parent, that is coming in the ovum. Another is by male parent, that is coming in sperm, and they are joined at fertilization randomly. These gametes have only one copy of each chromosome, are described as haploid. So gametes are having only one copy of the chromosome. So these all you have studied in meiosis. When I was taking the lecture of meiosis, I have explained that how these alleles are coming to the sex cell or heterosomes. Okay. Mendel's dominance states that in heterozygote, one trait conceal the presence of another trait for the same characteristic. So that is phenomena of dominance. So Mendel states that in his dominance phenomena, he states that in heterozygote only one trait will be expressed, another trait will be concealed by the first trait. Rather than both alleles contributing to the phenotype, the dominant allele will be expressed exclusively. So both alleles will not be expressing. Only dominant allele will be expressing, and it will hide the expression of recessive one. Now, the recessive allele will remain latent. Latent means hidden, but will be transmitted to offspring. But it will be there. The expression will be hidden, but it will be there, and it will be transmitted to offspring by the same manner in which dominant allele is transmitted. Means at the time of transmission there will not be any problem. That is called a law of segregation. So dominant and recessive allele will remain together without contaminating each other. They will not affect each other. However, expression of recessive will be concealed by the expression of dominant, but at the time of segregation or at the time of formation of gamete, both these alleles will be separating independent of each other. So the recessive trait will only be expressed by the offspring that have two copies of this allele. These offspring will breed true when self-crossed. So recessive only in case of homozygous they will be expressing. But I have already told you that we know today that the phenomena of dominance does not always hold true. Instead, several different patterns of inheritance have been found to exist. So what are those different patterns of inheritance? So we know the codominance, incomplete dominance, that I will be discussing in some other lecture. What is codominance, what is incomplete dominance. Now in nutshell, whatever things I have discussed today, you can see the tall plant is having allele capital T crossed with dwarf plant, that is small t. The capital T will be forming gamete having allele capital T. Small t will forming gamete having small t. So parents, gametes, and when these two gametes will fertilize, unite, they will form F1, that is heterozygous tall. The expression will be tall, that means tall is dominant. Tall trait is dominant. Now, if tall, this heterozygote is self-fertilized. Here you can see capital T small t crossed with capital T small t, that is self-fertilization. Then the gamete formed will be capital T small t, capital T small t. Now we can form the Punnett square. Punnett square is also known as checkerboard. Here we can check the male and female gamete combination. So male gamete capital T small t, female gamete capital T small t, and the progenies will be capital T, that is homozygous tall, capital T small t, homozygous heterozygous tall, then capital T small t, heterozygous tall, and finally small t, that is homozygous dwarf. And here you can see the ratio is 3:1 phenotypically. Phenotypically 3:1, that is 3 tall and 1 dwarf. But genotypically it will be 1:2:1. Why it will be 1:2:1? 1 will be homozygous dominant, tall. 2 will be the heterozygous dominant, tall, and 1 will be the homozygous recessive dwarf. So this is the genotypic ratio. So this is called as phenotypic ratio, and another one is called as genotypic ratio. So this is in nutshell what we studied today. So in next class we will be discussing about the dihybrid cross, and we will see how with the help of dihybrid cross law of independent assortment was given. So till then, thank you very much.